Hello everyone, it's my pleasure to introduce Gut-derived Lactobacillus from exceptional responders mitigates chemoradiotherapy-induced intestinal injury through methionine-driven epigenetic modulation. Acute chemoradiotherapy-induced intestinal injury severely affects treatment outcomes and quality of life with no established therapies. Addressing current microbiome intervention limitations, we developed an integrated approach combining fecal metagenomics and machine learning to identify ACRIII-associated microbiota signatures. Lactobacillus strains isolated from exceptional responders were validated in murine models. Lactobacillus rhamnosus DY801 emerged as a lead therapeutic candidate, demonstrating methionine co-metabolism-mediated efficacy through multi-omics analysis. This breakthrough provides both immediate treatment options and a microbiome drug development platform for ACRIII. Highlights: exceptional patient-derived lactobacillus strains, notably Lactobacillus rhamnosus DY801, effectively alleviate ACRIII. DY801 modulates host gut cystine and methionine metabolism. T cells are crucial pro-inflammatory mediators, the genesis of ACRIII. Microbial-derived methionine promotes histone methylation in T cells and reduces ACRIII-associated inflammatory responses. Post-treatment non-ACRIII patients exhibited higher relative abundances of Lactobacillus and Lactobacillus. Through the application of the random forest machine learning algorithm and evaluation using mean decrease in accuracy, Lactobacillus was identified as the most important genus in predicting ACRIII. 10 novel Lactobacillus strains with stable genetic characteristics were successfully isolated. Wild-type Lactobacillus strains, Lactobacillus rhamnosus DY801, Limosilactobacillus fermentum CYQ09, and Ligolactobacillus salivarius DY802, demonstrated robust overall performance in the in vitro lactobacillus screening experiments. L. rhamnosus DY801 significantly ameliorated ACRIII and suppressed both local and systemic inflammatory responses. DY801 effectively alleviated ACRIII without compromising the efficacy of in situ tumor treatment. Cystine and methionine metabolism was the crucial pathway for DY801 to alleviate ACRIII. DY801 alleviates ACRIII by regulating intestinal methionine levels in a MET-dependent manner. T cells played a pivotal role in the pathogenesis of ACRIII. DY801 and its metabolites affected both T cell quantity and function. DY801 and methionine inhibited the production of intestinal interleukin-17 alpha and interleukin-22 through in vivo experiments. DY801 derived methionine was taken up via sulfurd3A2 and converted to SAM by MAT2B, regulating T cells. This process increased H3K4 triamethylation levels at the interleukin-17 alpha and interleukin-22 gene loci, directly suppressing their transcription and reducing cytokine secretion. The SAM-induced alleviation of ACRIII was reversed by cycloleucine treatment. Clinical samples across 3 independent cohorts confirmed the association between low baseline fecal abundance of lactobacillus, reduced blood levels of methionine and SAM, elevated interleukin-17 alpha and interleukin-22, and the occurrence of ACRIII. Summary: in this study, we has identified 10 novel lactobacillus strains derived from exceptional responder patients, with 3 standouts: L. rhamnosus DY801, L. fermentum CYQ09, and L. salivarius DY802, showing remarkable efficacy in alleviating ACRIII. Most significantly, we've uncovered that DY801's therapeutic effect works through its MET-derived methionine, which induces H3K4 triamethylation-mediated chromatin remodeling in T cells. This epigenetic modification effectively suppresses pro-inflammatory cytokines interleukin-17 and interleukin-22, leading to clinical improvement in ACRIII. These findings establish a groundbreaking new therapeutic strategy, using patient-derived probiotics from exceptional responders as targeted microbiome therapy for ACRIII.